the paper talks about how we can understand resistance to a form of treatment for melanoma called immunotherapies. And these are treatments that have led to dramatic improvements in our ability to cure patients with metastatic melanoma. But what is also what's always been a kind of a mystery is why in some patients these treatments can lead to impressive and long-lasting responses, but in others the treatment doesn't work as well as we hope. And one of the ways to resolve this riddle is to take a group of patients who respond to treatment and compare their tumors to patients who don't respond. And that approach has been taken by a number of investigators around the world. But unfortunately, this kind of analysis requires a large number of patients in order to filter out those changes between the tumors that arise because of natural variation and don't actually matter in terms of treatment response. And so what this study did in collaboration with Dr. Ellie Van Allen's group, we decided to take a different approach. And in this study, we looked at patients who have numerous metastatic tumors in their body, some of which responded to these immunotherapies and some did not. And in these settings, we make a comparison between the two tumors in the same patient, the immunotherapy sensitive tumors and the immunotherapy resistant tumor. And what we found was that only about a third of the patients that we profiled had a known mechanism of resistance, but that in 3 of the 25 patients, we found mutations in 2 related genes, SEC23 and SEC24. And so the next step was to confirm that these mutations actually conferred resistance to immunotherapy. And that aspect of the project was undertaken by a postdoctoral fellow in my lab, Muad Benamer, and he addressed this by replacing normal SEC24, this normal SEC24 gene with the mutated one that we found in these resistant tumors. And when he did that, he found that the mutant gene abolished the ability of cells to turn on damage signals through a protein called STING. And this, in turn, led to the inability of the tumor cell to be recognized by a series of immune cells called T cells.